Okay, 5 seconds. Okay, let's see. These hydrocarbons and glucose and other nutrient molecules and use that energy hold within those electrons to make ATP. And today we'll deep dive in detail the complex and beautiful series of chemical reactions that all of your cells do to maximize how much ATP they are getting. ATP you should be familiar with from Friday. ATP stands for adenosine triphosphate. It is a nucleotide that is directly used for energy by your cells. ATP, again, I'm not going to go over ATP again, but main thing to know, has 3 phosphates, hence the name ATP, which stands for adenosine triphosphate, 3 phosphates. ATP can be hydrolyzed by removing this phosphate, breaking apart the phosphodiester bond. Doing so releases energy that your cell can use. So breaking apart ATP into ADP plus 1 inorganic phosphate releases energy. Removing phosphates or adding phosphates to molecules is generally done by enzymes. Can anyone remind me what is the type of macromolecule that are enzymes? The ends on ASC, correct, but what type of macromolecules are they? Proteins, yes, all enzymes are proteins. Asterisk, there are exceptions, we'll talk about that. You'll talk about that in genetics, but for now, all enzymes are proteins. And as your classmate said, as a rule of thumb, all enzymes end in ASC. Again, there are exceptions, but whenever you see anything that finishes on ASC, it's an enzyme, which is a type of protein. Okay, an enzyme, so when you remove a phosphate from a molecule, that process is called dephosphorylation. That is basically removing a phosphate from a molecule. And this is usually done by a type of enzyme called phosphatase. Just the type of enzyme that does this. Phosphatase will remove phosphates from a molecule. On the other hand, the process of adding a phosphate to a molecule is called phosphorylation. And the type of enzymes that does phosphorylation are generally categorized as kinase. These are enzymes that will add a phosphate to a molecule. So phosphatase do dephosphorylation, kinase do phosphorylation. Okay, before talking about cellular respiration, I need to introduce another concept that is critical to understand today's lecture. And this is 2 types of chemical reactions called the oxidation reactions and the reduction reactions. In an oxidation reaction, a molecule will lose electrons. We say that that molecule undergoes oxidation. That molecule is oxidized. Molecule lose electrons. Those electrons have to go somewhere. So this is usually coupled with another type of chemical reaction called a reduction reaction. In a reduction reaction, a molecule will gain electrons. These two chemical reactions take place at the same time. So we usually talk about an oxidation-reduction reaction, commonly known as a redox reaction. Electrons are removed from a molecule, which becomes oxidized, and electrons are added to a molecule, which becomes reduced. Electrons move from one molecule to another. Question: what is the electrical charge of electrons? Yes?
Negative.
Yes, correct. All electrons have a negative electrical charge. We discussed that in detail on lecture 2. Okay, so let me give you an example of a redox reaction. Here you have 2 atoms of sodium and 1 molecule of chlorine, so 2 chlorine atoms. You have a reduction reaction. This is better. So first you have an oxidation reaction. Each of these sodium loses 1 electron. Again, electrons are negatively charged. So if the sodium loses electrons, then they will gain a positive electrical charge. So 2 electrons have been removed, 1 from each sodium, and now these are 2 sodium ions that are positively charged. Those electrons have to go somewhere, so we have a reduction reaction happening at the same time. We have 2 atoms, and these 2 reactions, the oxidation and reduction reaction, happen together, and so this will be an example of a redox reaction. Does that make sense so far? Okay, I hope so. Okay.
Question.
Yes.
So, could you clarify one more time? Since electrons are negative, why does the sodium become positively charged?
It loses electrons. Oxidation, losing electrons. If it loses negatively charged electrons, then now it is more positively charged. Because this is neutral, because it has the same number of protons as electrons. Again, from lecture 2, I believe. Good question. So when something loses electrons, it will usually become positively charged. And when something gains electrons, it will usually become negatively charged.
Thank you.
Any other question? Okay, why am I talking about redox reactions in a biology course? Because electrons have energy. We talked last week about this Gibbs free energy concept. This idea that molecules, just by the location of electrons, have some amount of chemical energy. And so some electrons are what we call high energy electrons. Due to the location of these electrons, they have high potential energy. And so if a molecule loses those high energy electrons, they will lose energy. The potential of energy of that molecule will go down. If another molecule gains those high energy electrons, they will gain energy potential. And so when you remove an electron from a molecule, when you oxidize it, it decreases the potential energy in that molecule. On the other hand, when a molecule gains electrons, it gains more energy. Here you are converting another part of your cell. These are called electron carriers. easy to understand. Their job is to literally carry these high energy electrons from one molecule to another. Electron carriers can very easily be reduced, so can very easily gain electrons, and can very easily be oxidized, and meaning can very easily give up those high energy electrons to another molecule. There are several electron carriers in your body. We care about 3 of them for this class. Number 1 is a molecule known as nicotinamine adenine dinucleotide, known as NAD. NAD is an electron carrier. You have the oxidized form of NAD called NAD+. This is the oxidized form of the electron carrier. NAD+ can be reduced, meaning it can gain electrons, something like this. NAD+, 2 electrons and 1 hydrogen ions, making NADH. This is the reduced form of this electron carrier. Does that make sense so far?
So the electron carrier is NAD?
So it has 2 forms, NAD+, which is the oxidized form, and NADH, which is the reduced form. They are both electron carriers. The definition of an electron carrier can easily receive and give electrons. Does that make sense? Okay. So, NAD is synthesized by your body from vitamin B, which is why every single cell needs tons of NAD, which is why vitamin B is an important part of your diet. This is one electron carrier. A second electron carrier that will be relevant for today is FAD. FAD can gain 2 electrons and 2 hydrogen ions to become its reduced form, FADH2. And another electron carrier that will be important next week when we talk about photosynthesis is NADP+. NADP+ can undergo reduction, can be reduced to gain electrons. It will gain 2 electrons and 1 hydrogen ion to make NADPH. But we'll talk about NADPH next week. Any questions so far? Is anyone completely lost? Okay. So, going back to this cartoonish representation of cellular respiration, we are little by little gaining a few more details. The way the series of chemical reactions known as cellular respiration work, our body is taking glucose, other carbohydrates, fats, breaking apart the atoms that make up those macromolecules, taking away those high energy electrons into NADH and FADH2. Those electron carriers will then bring those high energy electrons to a specific part of the cell, specific part of the mitochondria, so the cell can use that energy to make ATP. There is a lot of conversion of energy, a series of very complex chemical reactions. And the topic of today will go step by step into the main parts of cellular respiration. So, In cellular respiration, cellular respiration is considered an aerobic process, meaning that it needs oxygen to take place. We will see that it's not 100% correct, but we can consider the whole process of cellular respiration aerobic, meaning it requires oxygen. Aerobic is the opposite of anaerobic, meaning a process that is anaerobic can take place without oxygen, in absence of oxygen. So, the very general overview of cellular respiration looks something like this. Your cells take as a reaction one glucose molecule. As a reminder, glucose is an hexose, which means what? 6 carbons. Yes, glucose is a hexose, so it has 6 carbons. You should all know its chemical formula: 6 carbons, 12 hydrogens, and 6 oxygen. And we also use 6 molecules of oxygen through a series of chemical reactions that we'll see today. At the end, you have 6 carbon dioxide as a waste or as a byproduct, 6 molecules of water, also a waste, and around 34 to 36 molecules of ATP. Of course, this is a very simplified overview. Cellular respiration looks something like this. There are 3 main steps of cellular respiration. You have step number 1, glycolysis, which takes place in the cytoplasm. Glycolysis is the only step which is anaerobic. Glycolysis will take place in the absence of oxygen. Step number 2 is the citric acid cycle, also known as the DCA cycle. Both glycolysis and the citric acid cycle will make a little bit of ATP, not a lot, we'll see a little bit, but the main thing, during glycolysis and the citric acid cycle, we are breaking apart glucose, taking out those high energy electrons, putting them into electron shuttles, and sending them to the last step of cellular respiration, the electron transport chain, which makes the most ATP during cellular respiration. Questions so far?
Wait, the last, you said, putting it in the shuttles and sending it to the
Electron transport chain, which is the ETC, electron transport chain, uses those electrons to make the most ATP. But we'll go step by step today. Okay, let's start with glycolysis. So what we know as glycolysis is a series of 10 chemical reactions, order, one reaction after the another. Each of these chemical reactions is catalyzed by a unique enzyme. Glycolysis takes place in the cytoplasm and does not need oxygen, which again is why we call this reaction an anaerobic reaction. Does not need, can take place without oxygen. This is the very, very general overview of glycolysis. You start with one glucose molecule, which has 6 carbons. Through a series of 10 chemical reactions, you will be investing 2 ATPs, using up 2 molecules of energy. So 2 ATPs, which are hydrolyzed into 2 ADP. But at the same time, you are reducing 2 NAD plus into 2 NADH and 4 ATP into 4 ATP. So, you are spending 2 ATPs, but you are making 4 ATPs and 2 NADH. Which is why we say that the net gain of glycolysis is 2 ATPs and 2 NADH. Again, distinction, you are making 4 ATP, but you are spending 2. So the net gain of glycolysis is 2 ATP. Does that make sense for everyone?
But what about for the NADH that also being used?
Sorry, Anna, can you repeat your question?
There's only 2 NADH is being made?
Yes.
There's nothing more than that?
Nothing more.
Okay.
The first step, there's more steps in cellular respiration. But yes, at the end of this series of chemical reactions, you're breaking down your 6-carbon molecule, your 6-carbon glucose into 2 molecules of pyruvates. Each one has 3 carbons. Does that make sense so far? Okay, go ahead.
The top part is the osmosis part.
The blue arrow.
Great question. I didn't use the same colors. This is just what you're making, and this is what you are using. Great question. This is the only oxidation reduction reaction. Here you have a dephosphorylation. You're removing a phosphate, and here you have 4 phosphorylations. Great question. It is not the same color scheme. Thank you for bringing that up. I'm just showing here, this is what you're spending, and this is what you are making energy-wise. Okay, now,
So basically, the arrows is like this arrow, this is what's going in, and this is what's going out.
yeah, so remember, this is energy.
Right, so this is what you're gaining, and then the blue arrow is saying this is what you're using.
The other way, this is what you're losing. You are, but, does, no?
It's just opposite in my head.
Okay, we can talk in breaks.
Question, Professor.
Yes, go ahead.
When you say that we have a net gain of 2 ATP, that means that it's because the 4 ATP we have that we are making, it will convert to ATP.
So you're making 4 ATP, you're producing 4 ATP, but you are using 2 ATPs at the same time.
Okay.
So net gain, if I bet money and I lose $2, but I win $4, my net gain is $2.
Yes, that makes sense.
Does that make sense?
I just because I saw that using the top, it has ATP and then ATP, I just saw 2 ATP instead.
Yeah. So basically, here in red, you are using up 2 ATPs, you're using up 2 molecules of ATP, and in blue, in that corner, you are making 4 molecules of ATP. That makes sense?
That makes sense. It's just that they're in opposite parts, so I got confused.
Okay. Does that make sense?
Yes, now it makes sense.
Makes sense for everyone else? Okay, now let's look actually to what happened during glycolysis. Now, you don't need to know all the name of the enzymes or all the name of the intermediary products, but if you are taking the MCAT or you are going to take biochemistry, you will be asked to memorize all of these steps. We are not there, we are not doing that, so you don't need to memorize all of these steps. So let's go over in a general way what takes place during glycolysis. Glycolysis again is 10 chemical reactions. Glycolysis can be separated in 2 sections. We have the first half of glycolysis, step 1 to 5. It is colloquially known as the investment phase. Why? Because that's where you are investing energy, you are using ATP. You are not gaining anything during step 1 to 5, you are investing energy. So, this is very complicated, so let's see if this works. Okay, this is a lot of cartoon. but it should be easier. So we start with a molecule of glucose. It's all of the same as in the slides, which, as your classmate reminded me, glucose has 6 carbons. Each of these little balls represent 1 carbon. Step 1 of glycolysis is catalyzed by an enzyme. You do not need to know its name, but you should be able to guess what it does based on the name. This enzyme is called hexokinase. So who can tell me very generally what will hexokinase does based on the name? Yes? Yes, it's a kinase, so it will add a phosphate somewhere. Hexo, or hexose, it's an enzyme that will add a phosphate to a 6-carbon sugar. So here you have a phosphate that has been added here. So this step 1 is irreversible. Step 1 is irreversible. That phosphate has to come from somewhere. So you have 1 ATP is used. The ATP is hydrolyzed into ADP. And that phosphate is added to glucose. Does that make sense so far? And this molecule is now called glucose 6-phosphate. You don't need to know the name. But you can guess what it is based on the name. Glucose 6-phosphate. Step 1 is irreversible because it requires energy. It's irreversible, so you can see an arrow going in one direction. Step 2 is reversible, meaning it can go, you have an arrow in both directions. Step 2 is catalyzed by an enzyme called an isomerase, which should remind you of the term isomer, which we discussed several weeks ago. Can anyone remind me what is an isomer? Yes?
It's like when a molecule has the same atoms in it, but it's
Yes, so when two molecules are isomers, when they have the same atoms, but the way the atoms are organized are different. So here you have the same general chemical formula, but instead of a glucose 6-phosphate, you have a fructose 6-phosphate. Fructose is an isomer, you don't need to know the name. It is just another sugar that has 6 carbons that also has a phosphate on carbon 6. Does this make sense so far? Yes? Can you pick up? From glucose to fructose? Step 1? 1 to 2. Yes, there is an enzyme, a kinase, that will take the phosphate from ATP and add it to glucose. That requires energy, and that energy comes from the hydrolysis of ATP. Breaking apart ATP into ATP requires energy. That energy is used to put that phosphate into glucose. Does that make sense?
That's your second ATP that you use.
There is no second, so you have a phosphate here. That phosphate doesn't go anywhere, so nothing
No,
has
what I'm
been used.
saying is that in this whole process, you say you're going to use 2 ATPs, and in the first step.
Oh yeah, there is more. There is more. There is more. We're not done yet. We are not done yet. Okay. Wait, question. So that's your question too? Okay. Next, we have step 3. So this is step 2. In step 3,
Question.
yes.
How does glucose become fructose though?
The enzyme just helps moving the atoms of the molecule around.
Oh.
You remember these are isomers, so they have the same atom, just the orientation of some of the atoms is different.
Oh,
So that doesn't require energy, which is why it goes in both directions. Does that make sense? Okay. Next we have step 3, which is irreversible. Step 3 requires an enzyme that's also kinase. This enzyme will take the phosphate from an ATP and add it to this 6-carbon sugar. So now from 1 phosphate, now we have 2 phosphates. And that is where the second ATP that is used comes from. 1 ATP was used in step 1, and other ATP is used in step 3. Step 1 and 3 so far are irreversible. This molecule, you don't need to know the name, but it's called fructose 1,6-biphosphate. You don't need to know the name, but you can guess what it looks like. 1,6-biphosphate, it has 2 phosphates, 1 phosphate in carbon 1, 1 phosphate in carbon 6. Does that make sense so far? Yes. Yes, sorry, these are keys. They're supposed to be 1 phosphate here, 1 phosphate here. Yes. Any other questions? Okay. This is step 3. In step 4, this 6-carbon molecule is broken into 2 molecules, 3 carbon each. This is step 4, which is reversible. So you have two molecules, 3 carbon each. One of each of these molecules have a phosphate. You don't need to know the name of this intermediary. That is step 4. Does that make sense so far? Okay, and step 5 is done by another isomerase that shifts, moves around some of the atoms in each of these 3 carbon molecules, making a molecule that still has 3 carbons and 1 phosphate. This molecule is called G3P, which stands for glyceraldehyde triphosphate. It's all the slides. Does anyone need more time with this cartoon?
Yeah, hold on.
Okay. So we are at the end of step 5. We have 2 molecules of G3P, glyceraldehyde triphosphate. Triphosphate because the phosphate is of carbon 3. Each of these molecules has 3 carbons and 1 phosphate each. And that is the end of step 5 of glycolysis.
Professor,
Yes.
you said that if something is reversible or not, so I'm trying to figure out.
You need to know that, but yes. 1 and 2.
Well, I understand that, but I'm trying to figure out what would reverse this process. If you take in ATP and making it ATP, so then the reversal would be to add a phosphate.
Which requires energy. So making this chemical, let's say this chemical reaction, requires energy. That energy comes from this dephosphorylation, which is why this reaction is irreversible. Does that make sense? Because going the other way, yeah, I don't know how to,
Yeah, you can't add the
yes.
phosphate
because that will also require energy, and then you will need, in theory you could if you have a special enzyme, but there is no special enzyme to do that. Does that make sense? Okay, great question though. Okay, so all of these in these slides. At the end of step 5, you have again these 3 carbon molecules with 1 phosphate, and this is glyceraldehyde triphosphate. 3 phosphate because the phosphate is on carbon 3. Okay, now we go in, so in this first half of glycolysis, you have taken 1 glucose molecule, which has 6 carbons. You have done 5 chemical reactions where you have spent energy from 2 ATPs in step 1 and step 3. And at the end, you have 2 molecules, 3 carbon each, and 1 phosphate each. And this molecule is called G3P, or glyceraldehyde 3-phosphate. Yes?
So for reversible sandwich losses would require a different enzyme?
It is the same enzyme. You can do both sides. That's a great question as well. Great question. Okay. So, we are now in the second part of glycolysis. Step 6 to 10. So far, in step 1 to 5, we have spent energy. We have used 2 ATP molecules, but we have not made any energy. Step 6 to 10 is known as the energy releasing phase. This is where the cell is at last making some energy. So you have some bunch of text here, so let me draw stuff for you. Again, we finish on step 5 with 2 molecules of G3P. What I'm going to show you right now is what happens with 1 molecule of G3P. For each glucose, we make 2 of these. So this is going to happen, what I'm going to show you next, happens twice. Again, I'm just showing you what happens for 1 molecule of G3P. So first, on step 6, which is reversible, something really nice happens. A high energy electron is removed from G3P and added to 1 NAD+. So NAD+ is reduced, making NADH. Oh my god, this, sorry. Okay. So again, 1 high energy electron is removed from G3P. G3P is oxidized. That electron goes to NAD+. NAD+ is reduced into NADH. This reaction is so energetically favorable. There is so much energy released when this happens that the cell can at the same time phosphorylate this G3P, add an additional phosphate, like this. Again, in reaction 6, NAD+ is reduced into NADH, and at the same time, 1 inorganic phosphate is added into G3P. You don't need to know the name of this intermediary molecule. It's called biphosphoglycerate. It's just a 3-carbon molecule with 2 phosphates. We are at step 6. Now we can start making more energy from here. In step 7, the cell, which is also reversible, the cell can take 1 of those phosphates with an enzyme and add those phosphates, one of those phosphates, to ADP, making ATP. And now we are back to a molecule with only one phosphate. Yes? Yes, in step 7? Yes. And I want you guys to tell me at the end if the cartoons are useful or not. I used to draw it, but it's very hard to draw in this computer. So at the end of step 6, we have a 3-carbon molecule with 2 phosphates. In step 7, the cell removes this phosphate and adds it to ATP to make ATP. And so at the end, you have a 3-carbon molecule with only 1 phosphate, very similar to G3P. Not exactly the same, it's called glycerate, 1-phosphoglycerate, I believe, but yes. Does that make sense? Okay, we're on step 7. Step 8, it's similar, is done by an enzyme called mutase. It's kind of an isomerase. You take the atoms that make up the molecule and you move them around, and you finish with a molecule that looks like this. Nothing has been removed, things have just been moved. 3 carbons, the phosphate has been moved here. This is step 8. And then step 9, also reversible. Atoms are moved within the molecule again. 1 molecule of water is removed out, but you still have 3 carbons and 1 phosphate here. This is done by an enzyme called phenolase. This step is also reversible. We're at step 9 right here. And the last step, in step 10, step 10 is irreversible. Kinase will remove the phosphate from this molecule and add it to ADP, making 1 more ATP. This is step 10. It's irreversible. You remove the phosphate of this molecule, you add it to ADP, and you make it phosphorylate ADP into ADP. And at the end, you have these 3 carbon molecules that is called pyruvate. In summary, step 1, 3, and 10 are irreversible. In the second half of glycolysis, for each of these G3P molecules, you made 1 NADH and you made 1, 2 ATPs. Does that make sense? Yes?
So, we create ATP or remove that those
With the exception of these step 6.
step 6.
Yes.
The
But generally speaking, most cases, losing an ATP is an investment of energy and will make it irreversible. Step 6 is a little bit of the exception because how favorable that reduction of NAD plus is. Does that make sense? Okay. So. Okay. So, this is what happens for each of these G3P molecules. Remember, this happens twice because for each glucose you make 2 G3Ps. Any questions so far? Go ahead. That is a great question. Your classmate asked, why are we doing all of this nonsense if we are not making energy and we're just moving things around? The reality is that, and this is far away from my field of studies, but evolutionary speaking, we have an enzyme that does this. We don't have an enzyme that can remove a phosphate from here. So you need all of these intermediary steps to reach something that you can remove a phosphate from. It is not ideal. I agree, you have so many steps that are kind of a waste of energy and time, but that is what our cell can do energy-wise. I said before that the cell is trying to be as efficient as possible. That's kind of not true. This process, all of these, what we'll talk today, is very inefficient. For each glucose that's broken down, your cell recovers only 30% of the energy in the form of ATP. The rest is lost in the form of heat, intermediary products, chemical reaction that did not finish. So even this, all of this happening step by step is the ideal case. In many times, the cell will take some intermediary and use it for something else. And the cell needs these intermediaries because it cannot do the shape of the molecule does not allow for these phosphates to be removed without all of these in between. Your question is good, why? And the reason is because that's the only thing that works for our cells. Yes? The what, sorry? This is G3P. You don't need to know the name of this molecule if you want it for your, it's on the slides. It is 1,3-biphosphoglycerate, but you don't need to know it. But you do need to know G3P, pyruvate, and glucose. Yes? You should know what comes in and what goes out. We separated glycolysis in two halves. What comes in, roughly what happens, and what comes out at the end of each half. This, in my opinion, is easier than just memorizing names and numbers, because you're seeing visually what happens, but if you can visualize the numbers, go ahead. But what comes in for each step, for each, not step, but for each section, or what comes in in the investment, what leaves the investment, how much was used up, and how much was made, if any, what comes into the second phase of glycolysis, what comes out, what was made, and what was used up. Any other question? Yes?
For step number 9, is that H2O that you're
H2O is removed, yes. Thank you. Okay, let's take a break. What are we time? 3:20. Perfect. We'll take a 10-minute break. We'll reconvene at 3:30. Are produced by one C. Okay, most of you technically got it right. Anyone tell me why is this the correct answer and not answer B? Why is C the correct answer? Yes? From the net gain from glycolysis. When you say net gain, you're taking into account whatever energy has been spent. Does that make sense for everyone? Okay. So, we did this through. So, so step 2 is we're not going to watch this video, but the link is there if you need help. So, I will also tell you that we go directly into the TCA cycle, but we don't. there is an intermediary step. So if there is oxygen in the cell, the pyruvate molecules that have been produced by glycolysis will move into the mitochondria, inside the mitochondria into the mitochondria matrix. And before these molecules can go into the TCA cycle, they need to be converted into another molecule. So we have an intermediary step that's called pyruvate oxidation. So we start with pyruvate, the 3-carbon molecule. During, again, pyruvate oxidation in the matrix of the mitochondria can only take place if there is oxygen. So this is a reaction that is characterized as aerobic, meaning it requires oxygen. Pyruvate oxidation is catalyzed by a complex of enzymes that is known as pyruvate oxidase. Yes, it's a complex of 3 enzymes and that do 3 different chemical reactions on pyruvate at the same time. First, this complex of enzymes removes one of the 3 carbons. This carbon is removed and is lost as a carbon dioxide. So this carbon here is removed out of this molecule. It is converted in carbon dioxide and is expelled out of the cell as a waste. So we go from a 3-carbon molecule to a 2-carbon molecule called acetic acid. Then one of those high energy electrons is removed from this molecule. This molecule is oxidized. This is coupled with a reduction reaction where NAD plus is reduced into NADH. At the same time, another part of the same end complex of enzymes takes a large macromolecule called coenzyme A and attaches it to this 2-carbon molecule, making a 2-carbon molecule called acetyl-CoA, which stands for acetyl coenzyme A. In summary, in this step of pyruvate oxidation, we go from a 3-carbon molecule into a 2-carbon molecule. That carbon is released from the body, from the cell, as carbon dioxide. At the same time, you're reducing one molecule of NAD plus into NADH, and you are attaching this coenzyme A to this 2-carbon molecule, making acetyl-CoA. And acetyl-CoA is the reactant of step 2 of cellular respiration. What does exactly this coenzyme A looks like? You don't need to know this. This is to show you how complex this whole process is. So these 2 carbons here is the acetyl group of acetyl-CoA. So these 2 carbons here are these 2 carbons here. And all of the rest here is the coenzyme A group. It is a quite a complex molecular group. It is synthesized from vitamin B5. Your body uses vitamin B5 to make this acetyl-CoA molecule. And acetyl-CoA goes into step 2 of cellular respiration, the citric acid cycle, also known as the TCA cycle. Any questions so far? Yes. Go ahead. Go ahead. So we have another three steps, glycolysis, PCA, and electron transfer chain. In between glycolysis and the PCA cycle, there is something called pyruvate oxidation. Like technically it's not glycolysis, technically it's not the PCA cycle. It's an intermediary step because the product of glycolysis does not go directly into the citric acid cycle. Does that make sense? Yes, but I just showed the three steps, that's the pyruvate oxidation. Great question. Yes? One of the products is citric, one of the intermediate steps is citric acid. PCA stands for 3-carboxylic acid, and they have another name, just a name. Citric acid is the name, I believe, because one of the intermediaries is citric acid, the molecule known as citric acid. Also a good question. Anything else? Okay, before entering into the citric acid cycle, a brief reminder of what the mitochondria looks like. How many membranes surround the mitochondria? Anyone? Yes? 2 membranes, yes. So there are 4 layers of phospholipids or 2 phospholipid bilayers around the mitochondria. You have the outer membrane, so that's in contact with the cytoplasm, and the inner membrane has a bunch of folds that looks like this. In between the outer and the inner membrane, you have the intermembrane space. Right, great. Just to make sure we are all in the same page. The pyruvate oxidation takes place in the matrix of the mitochondria, and then we have the citric acid cycle that also takes place in the matrix of the mitochondria. Again, you don't need to know all of these details. If you ever take the MCAT, you will need to know all of these details, but we are not there. So to try to I want to just emphasize a few things before we go into a simplified version of the citric acid cycle. Oh, citric acid, because here you have citrate as one of the products. Okay, so this is what we call in biology a cycle. This is called a cycle because, can anyone tell me why do they think this type of schematic is called a cycle? Anyone? Not scientifically accurate, but good, fine, acceptable. The scientific definition of a cycle is that you have in your cycle, the product, so what you have at the end of the cycle is the reactant. So, like your classmate says, it keeps going. Basically, what you have at the end of your series of chemical reactions will be reused for the beginning. That is the definition of a cycle. You basically end where you started. Alright, so again, this is a lot of detail, so we're going to simplify it a little bit with more of my drawings. So again, all of the information here is on the slides. I'm just going to show it as cartoon. Alright, we start with our 2-carbon molecule of acetyl-CoA. Acetyl-CoA again has 2 carbons, 1 and 2. The citric acid cycle begins by an enzyme. Each of these steps is catalyzed by an enzyme. By an enzyme taking these 2 carbon molecules and adding it to a 4-carbon molecule. This 4-carbon molecule is called oxaloacetate. Again, it's all on the slides with better writing, oxaloacetate. This enzyme will take the acetyl-CoA and fuse it with the oxaloacetate. These two together will be brought together to make a 6, so you have 4 carbon and 2 carbon brought together to make a 6-carbon molecule. that is called, you don't know the name, it's called citrate. So this is step one. You take your 2-carbon acetyl-CoA and you bring it together with a 4-carbon molecule of oxaloacetate together to make a 6-carbon molecule. Then, in a first chemical reaction, you are going to break apart, remove one of these carbons. When doing this, you are releasing a high energy electron that goes into NAD+. So this 6-carbon citrate is oxidized, it loses electron, and also loses a carbon. Those electrons go into NAD+ to make NADH. NADH. And that carbon is expelled as carbon dioxide. So you go from 6 carbons to 5 carbons, and you make so far 1 NADH. Does that make sense for everyone so far? I'll take that as a yes. Yes. You are here, 6 carbons. You have a chemical reaction where this molecule is oxidized, it loses electrons. Those electrons go into NAD+ to make NADH. At the same time, one of these carbons is removed, which becomes CO2. So you go from 6 to 5 carbons. Does that make sense? You're breaking apart that 6-carbon molecule, releasing those high energy electrons, which are going to NAD+. When you break apart, you're removing 1 carbon, which becomes carbon dioxide. Does that make sense for everyone so far? Yes, go ahead.
What you're explaining right now is a citric acid system?
Yes.
And you said that the NAD+ is being oxidized.
It's reduced, NAD+ is reduced into NADH. This is oxidized. And the NAD+ is reduced into NADH. The 6-carbon citrate is oxidized because it loses electrons.
Okay.
Thank you if I made that mistake. Any other questions? Yes, go ahead. This is one step. Yes. I'm simplifying it. You don't need to know intermediary. One step. For this class is one step. Okay. Any other question? Okay. Then this happens again. This 5-carbon molecule is broken down. Through an enzyme, one of these carbons is removed, which leaves the cell as carbon dioxide. So you go from 5 carbons to 4 carbons. And at the same time, this is coupled with the reduction of NAD+ into another NADH. Does that make sense so far? So far we've gone from a 6-carbon molecule to a 4-carbon molecule. We've made 2 NADH molecules, but we also made, as a waste, 2 molecules of carbon dioxide. That just leave the mitochondria, leave the cell. A lot of waste. Questions so far? Okay. Next, we do have a series of chemical reactions. Any questions?
Yes.
Go ahead.
Which step is reversible and which one is
Oxidation reduction. Oxidation is when you remove electrons. So in this case, you are removing you're removing electrons from this 6-carbon molecule. those electrons go into NAD+, so NAD+ is reduced into NADH.
No, I'm saying which one is the low?
Oh, what's a good question? I'm 100% sure the first one is irreversible. I am not sure which of these is reversible or not. But at least this one is irreversible. It's not as important to know which ones are reversible. That is a great question. Thank you. Any other question? Okay. Next, so we are here, a molecule with 4 carbons. We have a series of chemical reactions that I won't go into detail, but you're basically rearranging, removing, and adding chemical groups. We're going to stay with 4 carbons, but during this series of chemical reactions, a few things happen. First, you have phosphorylation of a molecule called GDP. Never heard about this before. There's a molecule called GDP that is phosphorylated into GDP. What does this make you think about? ADP into ATP. GDP is technically another molecule of energy that the cell can use. The cell can use ATP for anything. GDP can only be used for a few chemical reactions, but GDP is still technically a molecule of energy. Again, if you're confused, GDP plus phosphate becomes GTP. Guanosine diphosphate becomes guanosine triphosphate. It's green, GTP.
So is that energy specific to mitochondrial stuff?
This is a great question. This is made by the mitochondria, but the chemical reaction that can use GTP has nothing to do with the mitochondria. I believe I remember some of them for some enzymes can use GTP, if I remember correctly, but again, less useful, but still a molecule of energy. Great question. So that's one of the intermediary steps. In another intermediary step, the cell removes a high energy electron from these 4 carbon molecules. In another intermediary step, the cell removes a high energy electron from these 4 carbon molecules, which is then used to reduce FAD into FADH2, which is another electron carrier that we discussed earlier today. And in another last step, another couple of electrons are removed from these 4 carbon molecules to reduce NAD into NADH. And at the end, we go back to having these 4 carbon molecules. After all of these chemical reactions, we are back to oxaloacetate these 4 carbon molecules. And the cycle is ready to start again. Go ahead.
How many of these cycles happen in one
Great question. Great question. Each glucose makes 2 pyruvates. Each pyruvate becomes 1 acetyl-CoA, so you can do 2 of these cycles per glucose. Does that make sense? I encourage you to remember that, but also think about the logic. If you just try to memorize numbers, you will get confused. So it's fine if you remember the numbers, but also think about why is 2 of these per glucose. If that makes sense. There's a lot of numbers. If you just try to memorize the numbers without understanding why, you will get confused. Yeah, 2 of these cycles per glucose. So again, at the end of the citric acid cycle, you start with 1 acetyl-CoA, one oxaloacetate, all of these molecules that have no energy, and at the end, you regenerate the oxaloacetate. You've made 2 carbon dioxide as waste, but you've generated 1 GTP, 3 NADH, and 1 FADH2. Again, all of what I just explained, it's on the text of the slides. I just drew it because I think it helps see the step by step. Questions? Yes. Yes. For each pyruvate becomes 1 acetyl-CoA or 2 acetyl-CoA, and for each acetyl-CoA, the cycle happens twice. I'm sorry, for each acetyl-CoA, the cycle happens once. Does that make sense? Okay. Which brings me to my question. What is the net gain of energy? So ATP, GTP, and electron carriers produced by one glucose molecule in glycolysis, pyruvate oxidation, and the citric acid cycle. So I will give you a couple of minutes. You have the TA for help. This is the level of detail that you need to understand also for the exams, the intermediate steps, but also what comes out energy-wise. And again, the question is the net gain. Not what is produced, but what is the net gain. Okay, 10 seconds. Okay, so let's see. most of you got it right. Yay. Who wants me to go step by step? Okay, let's go step by step together. Okay, we start with glycolysis. Can I write this? Yes. In glycolysis, the question is asking you from one glucose. At the end of glycolysis, what happened to that glucose? Anyone? No, just what happened to the molecule of glucose? Two pyruvate, correct. Okay. Then the question is asking you for the net gain. I heard someone said 4 ATP. That is not the net gain of glycolysis. 2 ATPs. Why? You make 4 ATPs, but you are using 2. The production of ATP in glycolysis is 4 ATP. The net gain is 2 ATPs. Does that make sense? Yes? Yes, okay. So 2 ATPs. And how many? And 1 and 2 NADH. Does that make sense for everyone? Okay. Next, you have pyruvate oxidation. From one pyruvate, you transform it into one acetyl-CoA. During this process, you reduce NAD plus into NADH, but this happens twice because you have 2 pyruvates, so you get 2 NADH out of pyruvate oxidation from the products of glycolysis from one glucose molecule. Does that make sense? Okay. So at the end, you have 2 acetyl-CoA. Okay. Then you have the TCA cycle. In the TCA cycle, for each acetyl-CoA that enters, you make 3 NADH, 1 GTP, and 1 FADH2. Now I just told you, at the end of pyruvate oxidation from one glucose, you have 2 acetyl-CoA. So you multiply all of these by 2. So you have 6 NADH, 2 GTP, and 2 FADH2. If you were to add all of these, you have 2 ATPs, 10 NADH, 2 FADH2, and 2 GTP. Does that make sense? Anyone lost or confused? You need to be able to do this the day of the exam with any of the intermediaries I've told you during this. The main intermediaries, not the middle of the road, but during glycolysis, during pyruvate oxidation, what comes in, what comes out. You need to know what are the energy products for each of these main steps. Questions?
Professor,
Yes, first.
for Rouvier oxidation, what is that that says underneath it? Do what?
For each pyruvate, you make 1 NADH. Because for each glucose, you make 2 pyruvates, so you're making 2 NADH. Does that make sense?
Yes.
Okay, yes. Can anyone tell me why I start multiplying? Someone. Yes. You got? Yes, you do it twice because for each glucose you make 2 pyruvates, and after that I told you what happens for each pyruvate. So if you're looking at just what comes from one glucose, you multiply everything times 2 because you are making 2 pyruvate molecules from one glucose. Does that make sense? I don't know what's the question. Hope that makes sense. Yes. I messed up somewhere. 2 times 3, 6, 8, 10. It is 10. Am I wrong? 2 times 3, 6, 8, 10. No? Did I mess up some? Okay. Okay. Any other question? Yes. Yes, which is why I multiplied everything by 2. Each cycle of the citric acid cycle turns with 1 acetyl CoA. From glucose, you have 2 pyruvates. From 2 pyruvates, you have 2 acetyl CoA. So the citric acid cycle turns twice. And yes. Yes. We go to my ugly drawings. All of this happens with 1 acetyl CoA. From glycolysis, 1 glucose becomes 2 pyruvates. 2 pyruvates become 2 acetyl CoA. And so for each acetyl CoA, we do that cycle once. Which is why I multiplied all of the products times 2. Because you have 2 acetyl CoA for each glucose. Does that make sense? Any other question? Okay, great. So, yes.
We have a question.
So I'll go where?
Here. Up here.
Oh, yes.
Why didn't you add the 4 ADP for glucose?
Why did that?
Why didn't you add the 4 ATP for glucose?
So here?
Yes.
The question asked for the net gain. The net gain is after you take out whatever you spent already. So for example, if I were to gamble and I lose $10 but I won $15, even though I won $15, my net gain is $5 because I spent 10. Does that make sense? So the question is, what is the net gain? In this case, the net gain from glycolysis is 2 ATPs because you produce 4 ATPs, but you spend 2 of them during glycolysis. Does that make sense? Yes. Okay. Any other question? Okay. Moving on to the last part. Okay. So far from this question, you know, with only the net gain so far is 2 ATPs and 2 GTPs, which is not a lot. On the other hand, the cell has produced 10 NADH and 2 FADH2, which is a lot. In the last step of cellular respiration, the electron transport chain, the cell will use those reduced electron carriers and use their energy to make ATP. How does that work? Basically, the last step of cellular respiration is a series of processes that is called the electron transport chain. During the electron transport chain, special proteins and molecules will receive those high energy electrons and will pass them around. Each time those high energy electrons move from one molecule to another, a little bit of energy is lost. That energy will be used by the cell to make ATP. This is summarized in this cartoon. So you have NADH and FADH2 carrying these electrons that are full of energy, high energy electrons. These high energy electrons are given to the electron transport chain, and the electron transport chain will pass around those electrons. Every time they lose energy, that energy is used to make ATP. At the end, you have electrons that are useless. They have no energy whatsoever, and they need to go somewhere. Here comes oxygen. Oxygen is considered the ultimate electron acceptor. 2 electrons will be given to half of a molecule of oxygen. 2 electrons plus half of a molecule of oxygen, and 2 hydrogen ions to make water. This is what you see here. And so oxygen is considered the ultimate electron acceptor because it receives those useless, energyless electrons, taking them away so the process can continue. What does this process look like? So this is what the electron transport chain looks like. There's a lot here to see. We are in the mitochondria. And as a reminder, this is the inside of the mitochondria matrix. As explained earlier, the mitochondria has two plasma membranes. We have the inner membrane, and we have the outer plasma membrane. And in between the outer and the inner membrane, we have the intermembrane space. Does that make sense so far? Okay, I assume yes. So, a few things to note. We have here a concentration gradient of hydrogen ions. We have a lot of hydrogen ions here in the intermembrane space. And we have very few hydrogen ions here in the mitochondrial matrix. And also, another thing to note is the different components of the electron transport chain. We have here a protein called ATP synthase. Just based on the name, what can you tell me about this protein? It's an enzyme, yes, which makes ATP. If you want to guess, but you should be able to know, based on the name at least, that it's an enzyme. This ATP synthase, it's an enzyme that makes ATP. It is also a protein channel that will allow diffusion of hydrogen ions by down their concentration gradient from high to low concentration. I will explain ATP synthase in a second. There are also other enzymes, other molecules. You have complex 1, 2, 3, 4. You have here a molecule called coenzyme Q, and you have a molecule here called cytochrome C. All of those names are on the slides, on the text slides. Complex 1, 3, and 4 have the ability of receiving electrons and passing those electrons to another molecule. And they are also hydrogen pumps. Can anyone remind me from Friday what are pumps in biology? Active transport, yes. Pumps move molecules by active transport, so from low concentration to high concentration. Complex 1, 3, and 4 are hydrogen ions pumps. They move hydrogen against their concentration gradient. Complex 2, coenzyme Q and cytochrome C they just receive electrons, high energy electrons, and pass them around. All of that information is, and so this is the electron transport chain. All of that information in text is here. Yes. So now, what exactly takes place during the electron transport chain? And for that, I have a cartoon I'm going to draw into. Okay, this is a lot. So first, you have ATP synthase. You can think of ATP synthase like a water wheel or a windmill. ATP synthase is basically a channel that allows hydrogen ions to move down their concentration gradient. When hydrogens cross ATP synthase turns like a motor, like a windmill, creating energy that is used by the enzyme to catalyze the phosphorylation of ATP into ATP. So, hydrogen ions will move down their concentration gradient from the intermembrane space, the area of high concentration, down ATP synthase to the area of low concentration. This causes ATP synthase to literally turn, creating energy that is then used for this chemical reaction. Does that make sense so far? If there is nothing else here, what will eventually happen to the concentration gradient of hydrogen ions? If there is nothing else to, yes?
It will like neutralize the all of them will go.
Yes, the term is equilibrium, but you are correct. Your classmate says neutralize, but basically eventually you will have more and more hydrogen ions here until eventually there is no gradient anymore, which will stop ATP synthase and we will die because our cells need ATP every single second. So to prevent that, we have the second part of the electron transport chain here. So far, through the citric acid cycle and glycolysis, we've made NADH. NADH carries these high energy electrons. These high energy electrons will be passed from NADH into complex 1. Complex 1 receives those high energy electrons, becomes reduced. Complex 1 becomes oxidized and gives up those high energy electrons to coenzyme Q here. Every time high energy electrons are moved, they release a tiny bit of energy. That energy is used by complex 1 to pump hydrogens against their concentration gradient from the matrix out into the intermembrane space. So even though you have all of these hydrogen ions constantly moving down here, they are pumped against their concentration gradient here. Does that make sense so far? They said no, maybe, yes? So hydrogen ions are going down. At the same time, the energy from the electron of NADH is used to move this hydrogen out. So you always have that concentration gradient of more hydrogens here than here. And that energy comes from moving that high energy electron. That high energy electron is given, taken away from NADH, given into complex 1. From complex 1, that high energy electron goes into coenzyme Q. Nothing happens, coenzyme Q is kind of useless. But coenzyme Q receives that high energy electron and passes it down to complex 3. Complex 3 passes that high energy electron to cytochrome C. When it does so, a little bit of energy is released. That complex 3 uses to pump hydrogen out into the intermembrane space. Cytochrome C now carries those high energy electrons and they are passed into complex 4. Complex 4 receives that high energy electron that now has almost no energy and uses it, whatever is left, to pump hydrogen against its concentration gradient. Now those electrons have no more energy, they're useless, but they have to go somewhere. And so they are given to oxygen to make water, H2O. Which is why again, oxygen is considered the ultimate electron acceptor. And so complex 4 gives up those electrons, has been oxidized, so it's ready to accept more electrons from cytochrome C. If you remember, through the citric acid cycle, we not only make NADH, we also make FADH2. FADH2 is given, FADH2 gives their high energy electrons to complex 2. Complex 2 passes those electrons to coenzyme Q. Coenzyme Q passes those electrons to complex 3. Complex 3 uses that energy to push out, pump the hydrogen ions against the concentration gradient. Complex 3 passes those electrons to cytochrome C. Cytochrome C passes those electrons to complex 4. And complex 4 uses that energy to pump hydrogen against its concentration gradient. And the same, the energy is spent. It goes into oxygen to make water. Okay, and so that is how your cells make most of its energy. Basically, again, ATP synthase acts as a windmill, as a turbine. The hydrogen ions move down their concentration gradient, creating a physical force that the enzyme uses to make ATP. The last two slides before the workshop, I already explained this. Yes, this is a summary of what I just explained. But basically, ATP synthase from all of the products made from one glucose can create 30 to 32 ATP molecules just by the electron transport chain. So the vast majority of ATP is produced by the electron transport chain. All what I explained, last two slides before the workshop. So, again, total glycolysis, citric acid cycle, and electron transport chain, you make 30 to 36 ATPs per one glucose. 30 to 34 of those ATPs come from the electron transport chain, the vast majority of those ATPs. This is a very inefficient process. Only around 34 of the energy of glucose is converted into ATP. The rest is usually lost as heat, as a byproduct. Also, what I show you so far is this idealized case study where every single molecule goes into the next step. In reality, in many cases, intermediaries will be used by other chemical reactions. Okay, we'll finish this chapter next week, and then we'll do photosynthesis next week. Let's do today's workshop.
I wasn't even, I was lost after 18. Oh, I know what you're talking about. I just don't trust myself. I feel like if I had that in the fridge. Oh wow.
I'm sorry, what's the group situation here? Okay, just wondering. Because I know it's 5, right? Each group. left hand card.